In this video, we're going over the background for Lab 12, which is titled Qualitative Analysis for Carbohydrates. The goals of this experiment include that students will be able to recognize the different types of carbohydrates, which include monosaccharides, disaccharides, and polysaccharides. And students will be able to understand how chemical tests are used to identify carbohydrates. First, let's discuss what are carbohydrates. Carbohydrates are polyhydroxy compounds that either have an aldehyde or a ketone and contain 3 to 8 carbon atoms. These biomolecules can be further categorized depending on the amount of sugar units that they have. In the case of a monosaccharide, which I'm highlighting in yellow, as you can see, examples of monosaccharides include glucose, fructose, and galactose. And these are one of the most common monosaccharides. So even though we have 3 samples, only in this experiment, glucose is the one that is going to be studied. When it comes to disaccharides, here we have the examples of sucrose, lactose, and maltose. And in this experiment, we are going to be exploring and observing the results of sucrose and lactose. The difference between these 2 disaccharides that are composed of, in the case of sucrose, glucose and fructose, and in the case of lactose, by galactose and glucose, is that sucrose specifically is bonded at the anomeric position. So the 2 sugar units are bonded at the anomeric position compared to lactose, which is not. And we are going to see how the chemical test can identify those 2 different types of glycosidic bonds as a result of the characteristics of the disaccharides. Lastly, when it comes to the polysaccharides, we're only going to be observing what are the chemical tests doing in order for us to identify starch, which is a polysaccharide that contains many glucose subunits. Understand that starch, similar to sucrose, is linking many of these single glucose units, okay, together, and in this glycosidic bond that is unifying the glucose, we do not have an available anomeric position, and we are going to observe how that affects the results in the chemical test. Now, when it comes to the monosaccharides, as explained in the first bullet point, we are going to be categorizing specifically the monosaccharides according to the carbonyl functionality, meaning aldehyde or ketone, or the number of carbons. So when it comes to these monosaccharides, if they have an aldehyde group in their structure, they're going to be known as aldoses. While if your monosaccharide has a ketone group, then it's going to be known as a ketose. You can observe from the examples that we have here for erythrose and erythrulose that in the case of erythrose, we have an aldehyde functionality. That's why it's known as an aldose. While erythrulose has a ketone functionality, and that's why it's known as a ketose. The number of carbons present in our monosaccharides can also be utilized in the classification. And as you can see here, we have the example of glucose and fructose. Each of these 2 monosaccharides contain 1, 2, 3, 4, 5, 6 carbons. And since they have 6 carbons, they are known as hexose. Now understand that the aldehyde and ketone group that may be part of the molecule can be also be incorporated into the categorization for your monosaccharide. So if you have an aldose sugar, then the aldose sugar before the word hexose, pentose, tetrose, or triose, which determines the number of carbons, you can put ALDO for an aldose sugar, or KETO for a ketose sugar before the name for the number of carbons in the structure, and that's how you incorporate both classifications in one name. It is important to note that when it comes to carbohydrates, specifically when it comes to breaking them down, only disaccharides and polysaccharides can undergo breakdown. Now, what that means is that through the process of hydrolysis, which means cutting a molecule with water, if you have a disaccharide, that is going to produce each one of the monosaccharides that make that molecule. So for example, if I'm talking about lactose and it undergoes hydrolysis, that means that it's going to produce galactose and glucose. If it comes to polysaccharide, and in this course we only observe polysaccharides that are made out of glucose, we're going to make many monosaccharides that are the ones that made that polysaccharide. So in the case of having starch undergo hydrolysis, we will be making glucose molecules. There are a variety of chemical tests that can be utilized in order to identify if a sample have carbohydrates or is very specific in the type of carbohydrate that is in there. So in this experiment, you guys will be doing 5 different chemical tests. And the first one is the Molisch test. As you can see in the left side of the slide, these are the chemical reactions that are going to happen in the test tube in order to identify a carbohydrate sample. So the logistics of the Molisch test is that if you have a monosaccharide, a disaccharide, a polysaccharide, any carbohydrate, when you put it in the presence of sulfuric acid, that molecule is going to dehydrate and then it's going to make a furfural molecule. So in the example here, we have specifically the furfural that is going to be made from glucose. Then that furfural molecule is going to react with alpha naphthol. And naphthol is going to be a reagent that it is inside the Molisch reagent. And when that happens, understand that this is going to overall dehydrate again, and there's going to be an oxidation process in which a purple violet color complex is produced. So you know that your sample has any carbohydrate, monosaccharide, disaccharide, polysaccharide, as long as your test tube creates this violet colored ring. The next test that you guys are going to be performing is the Fehling's test. In the Fehling's test, what is going to be happening is that the aldehyde that is going to be in the carbohydrate, and you have 4 different samples of carbohydrate that you're going to be testing. If in the Fehling's test, that carbohydrate is changed into a carboxylic acid, then we know that that process of oxidation will result in the reduction, in this case, of a copper compound. So we're going to go from copper 2 to a molecule that it has a copper 1 oxidation number. So overall, in the Fehling's test, what is going to happen is that if you have an aldehyde that is able to undergo oxidation to the carboxylic acid, you're going to see that is going to produce a reddish precipitate. You will see that in the simulation, sometimes it looks like red orangey or even bright orange, but that is just the color that in the simulator they selected for it. So understand that any carbohydrate that is able to go from the aldehyde to the carboxylic acid, it is known as a reducing sugar. Similarly, in the Benedict's test, we are going to be observing a similar reaction. If you have a particular carbohydrate that is able to undergo oxidation and make that carboxylic acid, or in the case of the Benedict's test, make a carboxylate, and in turn is going to be converting that copper plus 2 to copper plus 1, then understand that this is also known as a reducing sugar. In the Benedict's test, similar to what we observed in the Fehling's test, is a transition from blue to orange reddish product. The test that also test for a reducing sugar, which is the Tollen's test. In this case, our reduction is going to happen at the level of silver. When silver gets reduced, in this case, is going to make silver in solid form. So similar to the two tests that I mentioned a few moments ago, if your carbohydrate is able to have its aldehyde functionality converted to the carboxylic acid, that means that is going to in turn reduce the reagent in this case that is going to be silver. But in the Tollen's test, because we are precipitating silver, then understand that we're going to see a dark gray precipitate or a silver color. But the Fehling's test, the Benedict's test, and the Tollen's tests are all tests for reducing sugars. Lastly, we have the iodine test. In the iodine test, this is specifically for starches. So if you have a starch in your sample, then understand that the transition is that since it is known that iodine is going to be reacting or interacting with the helix that is present in starches, that interaction creates a blue-purple complex. So if there's a starch in your sample, the transition from starting in the brown-yellow area to then form a product that is blue-purple, that identifies that a starch is present in the sample.